Okay, so it's my pleasure to introduce Donna Pierre as our first keynote speaker. She's the chair of the Computational Systems Biology program at Sloan Kettering Institute and a Howard Hughes investigator, well-deserved, recent investigator. So Donna got background was at Jerusalem, Hebrew University in Jerusalem. I think kind of was there for a long time. It's like, from seventh grade through, like mathematics, computer science, etc. So was there for quite a while with a computer science background. But I think there for graduate school, you also started on your biology interactions with VREG, etc., which helped kind of guide path forward. And then went to George Church's lab at Harvard Medical School for postdoctoral training, which really kind of further set you along path.
And where I met you.
Where we met the first time. I'll actually say something about that in a second. And, really got started much more using computational methods to answer real biology questions instead of computational questions being used. Biology is a tool. It's just a platform for doing computational stuff. I think my most striking interaction with Donna was actually the day I interviewed in my department for my job. And it was actually the same day that Donna was giving theory lunch in her department. So, no one was in my talk today. So instead of like the lunch with the speaker, they're just like, just go to her talk. And then, I was talking about doing using yeast variation between yeast in order to learn things quantitatively without signaling. And it's like, go to Donna's talk, like, it was, but they hired me anyway. Maybe they thought, pity. But anyway, so,
I never realized that I dampened your job talk, but you got it anyway. So
it was definitely fun. It was pretty amazing to see what you're up to at the time. And then after that, it's just been a great career of like starting with yeast and then applying those tools broadly outward. And then I think as many of us done, but you were a leader in really both taking advantage of things like single cell sequencing approaches and other things to apply these to human cells and cancer and disease and really start to take like lessons that we learned in yeast from variation, et cetera, to actually get towards more like genomic medicine, drug interactions in individuals. And so hopefully here.
Yeah, now my favorite organism is mice. They're more fun than human.
But it's been an incredible trajectory, and we're looking forward to hearing more about it from you. So thank you.
Yeah, thanks so much. Though you made me a somewhat evil person for ruining your or dampening your job talk, it's a pleasure to be here. Actually, one of my first talks as faculty was at one of first Cold Spring Harbor talk was at one of these the first one back in 2008. The field was very different then. My little thing sort of decided to fall off my pants, so I don't feel like holding my mic, so let me just fix this for a second. So, yeah, I've been, playing with G networks since my PhD. My first PhD project in ancient 1999 in prehistoric ages was applying microarrays to yeast to find G networks. During my postdoc, the whole idea of how we can actually derive molecular networks from single cell data at the time, just multidimensional flow. But recently, my biggest passion is not just looking at networks within cells, but looking at networks across cells and within tissue. So, before we delve into a specific example, I sort of wanted to give you the origins of how I really got passionate about this. I mean, it's obvious that we need to look at the level of a tissue. I mean, I'm not like having a deep insight here, but when I moved to Sloan Kettering from Columbia and encountered a few biological problems, it really sort of framed the way I'm thinking about it. And most of the projects in the lab really, look at two axes. So you have sort of the intercellular networks that compute how a cell will respond to its environment. It's something that we as a field have learned a lot more of, but still have a lot to learn. And then these intercellular networks sort of respond to their environments, creating emerging properties. And sort of that interplay between cell intrinsic and cell extrinsic environmental interaction. And now you can think of networks, gene networks within cells communicating through signaling molecules, creating gene networks across cells, and really understanding how this works in healthy systems, development, regeneration, and how cancer hijacks it is what I like to do. So, one of my earlier projects with Kat Agentinakis, looking at early embryogenesis, at how the gut tube forms, the endoderm, which really patterns all our main organs. And one of the things that Kat discovered is that extra embryonic tissue that's supposed to go become your yolk sac and your placenta, and not supposed to generate your body, during gastrulation, when the sort of gut tube forms by, turning inside out, as you see in this video, which just finished, sort of the extra embryonic cells sort of get sucked in and get intercalated here into the gut tube, which is sort of like a mosaic of embryonic and extra embryonic cells. And the question that we started out with is, what happens to these extra embryonic cells? What are the green cells doing? And single cell genomics was fairly new at the time, and we were lucky because 10x Genomics decided to fund this and sequence like crazy. But what we discovered is actually that these extra embryonic cells that a day before were completely crazy different, suddenly, once they intercalated into the gut tube, actually created like 99% identity with their adjacent cells. So if you're an extra embryonic cell near the liver, you're going to express cells like the epiblast, like the embryonic cells that are expressed in the liver, including the master regulators. And there's more than 99% identity. In fact, there's only 5 genes that differ, that sort of indicate the extra embryonic origin. There's no different genes in the epiblast. And so basically what we see is, these cells were about to become extra embryonic, and suddenly they look around and they're like, hey, all my neighbors, all the people around me are about to be pancreas. I have the innate plasticity to change course, change my entire transcriptional program, and become pancreas, which really shows you the degree of both internal plasticity that many cells still have, particularly during development and regeneration, and the degree in which extracellular cues can really impact you to change such a big phenotypic change. The other really, thing that really impacted the way I was thinking was work with Joanne Massaguet, with Ashley. And basically we were looking at metastasis and trying to look at what happens in lung metastasis. And looking at lung metastasis, the pattern that we found is really that the cells sort of organize into sort of like a pseudo-time that really matches, step by step, all the master regulators and all the targets in the correct order of actually building a lung. And what metastasis seems to be doing is trying to build a lung, sort of, it's messed up, it doesn't work, at the wrong time, at the wrong place, again, with massive plasticity and extracellular cues. So now most of the projects in my lab focus around these themes, and what I'm going to try and do here is tell, one story in depth, filling in a lot of the computational details, and if we have time, we'll go into another story. I usually pack my talks and then decide how much to talk and make you feel like you missed out a lot of my slides. So this is, my partner in crime is Scott. He's down the hall. We started having tea hour and, projects between labs metastasize, but it's really fun to have Scott down the hall, and we have a lot of joint shared similarities, but different skills. This was a sort of hand-in-hand, side-by-side project. They met every day, Cassandra and Dorena. Dorena is now faculty at IRB, and Cassandra is now a postdoc in my lab because she has just too many things going that she didn't want to leave after her PhD, and she's so talented that who can say no? And so we really wanted to understand the initiation of pancreas cancer. I mean, by the time pancreas is usually late stage, by the time PDAC is discovered, it's too late. And there's, most of the data, most of the studies of PDAC is advanced. So you have this, malignant, messed up, horrible, metastatic nastiness up here, and that's where most of the studies are. But we wanted to understand what happens early up here, where there's just one driver mutation, typically KRAS, but these cells look normal. I mean, there's a lot of studies that show, for example, that if you, sequence our skin, our eyelids, and all sorts of things, we have a ton of oncogenic mutations. I'm a walking bag of oncogenic mutations, and thank God, hopefully I don't have cancer. And so, what happens in this pre-malignant stage between the first driver mutation and the next driver mutation that pushes the cell towards cancer? Since we can't get, human samples, as I told Mike earlier, my favorite model organism is mouse, and you'll, in this project, see a little bit of why. And so with mouse, you could build all these powerful genetic models and, query whatever time points you want. And this particular genetic model has, the KRAS mutation specifically in acinar pancreas cells. It's also labeled so that we can enrich for them. They're pretty rare cells. So, if we would have just done vanilla sequencing without sorting, these would be 3% of our population, and we wouldn't, capture all the heterogeneity. So the ability to sort and enrich for rare cells is great, as well as the ability to induce cancer at a certain time and place. So we wanted to compare normal regeneration of the wild-type mice, because I really believe that you have to understand normal to understand disease. You can't just study disease out of context. And we did injury, so normal injury and regeneration, and we looked at injury, so early injury in the KRAS oncogenic setting, as well as what happens during neoplasia a few weeks later after the injury resolves, but not completely because it's oncogenic, all the way to after P53, what, malignant cells looking at the full gamut. So, here's the map of single-cell RNA-seq, the typical t-SNE that you see labeled by stage. What I want to show you here is, first of all, these things overlap. We have very many rare cells, and, this overlap is actually quite nice. And what we saw is that, even though these are different mice, at the very early stages, mouse after mouse after mouse, replicate after replicate after replicate, the early stages are canonical. Once you get to late cancer, it's not. But the first steps of towards malignancy, at least in PDAC, are very stereotypical and repeat itself. Now, when I look at data, this is a mess. It's hard to sort of dive into this sort of think about 80,000 cells in here with different types and some of them quite rare. So I like looking at the first diffusion component. The first diffusion component gives you, this is the strongest signal in the data. And since we were interested in tumor progression, it was nice to see that from the, normal pancreas to the regenerating pancreas, all the way to the really malignant pancreas, the first diffusion component really follows this axis of progression. Early on, in early injury in the malignant thing, we could see that these are more cancer-like than the normal, but we see a huge heterogeneity of cancer programs, EMT programs. So even though you see a lot of cancer-like programs activating in the early cells, it's done in a very heterogeneous manner. So because the first principal component is progression, a better way to look at the data to organize it a little bit better is using a diffusion map. So here we see sort of the early pre-malignant cells and the malignant cells. So we really wanted to sort of focus what's going on in the pre-malignant cells. And, one of the first questions we asked is, what are the initiating states? Because we sort of know that for development, but for cancer, the cell of origin, there was a big debate. The field's arguing this type of cell, that type of cell, lots of different lineage tracing pointing to different types of cells. Now, here's where pseudotime fails, because pseudotimes assumes that we know the starting cells. And it also assumes that cells go forward and don't differentiate. We know that both those assumptions are broken in regeneration. So one of the approaches out there that have been, developed to sort of, mediate that is RNA velocity that looks at splice versus, unspliced to try and see, what RNA is being made and where the cell might be going into the future to get some directionality directly from single cell data. It was a big splash, method of the year for a couple years ago. But as people began to use it, they found that it has many problems. Not only is it limited to an 8-hour window, so don't try and connect, 2 time points that are 5 weeks apart, as I see in many papers that I review, but it's also incredibly noisy. To show you how noisy it is when you look at these individual vectors rather than at these stream plots that typically average 1,000 cells at a time so they look nice, let's look at the degree of noise. Every single cell that's nearby and similar is pointing into a completely different direction. Many of the velocities are pointing out of the phenotypic manifold into future states that haven't been measured and don't exist. So while it does have some valuable information about directionality, RNA velocity on its own doesn't have enough oomph, and you certainly shouldn't trust these over-averaged heat maps. So this is, my lab loves to host people. It's the most fun thing. Science is all about collaboration, so I love this networking idea at the beginning. You come out of here with a collaboration that's more valuable than any science you might have heard from anyone at the meeting. And so, Marius came over from Germany to hang out with us for about 8 months, and we developed CellRank, which again is published in full gory detail. But now I'm going to do something. I'm going to test your how awake you are after a hard travel here at 8 p.m. And this biological story is going to have lots of different computational segues, so you're going to have to remember where I left off as I tell you a little bit about CellRank. So the idea of CellRank is, okay, we have some real directional information from the RNA velocity, but, the pseudotime have been proven to be very robust methods. They're global. They look at global things. They stick to the phenotypic manifold. So let's marry the two. And basically what we try and do is infuse pseudotime-like methods and methods that use a Markov chain and inside the manifold, but give them some directional information. So cells can only move within the KNN graph. They can only move to their neighbors, but the probability that they'll move to one neighbor or another is dependent on how aligned they are, how much they match the direction of the velocity. So again, it's a Markov chain. You see where it's going to go, and that probability is basically defined you're more probable if you're sort of in the direction of the vector. The nice thing here, though, is it's global. We have this Markov chain for each one. The vector infuses some directionality information, but now we do a global calculation. This was a lot of fun, too, because we actually used some very powerful statistics because, Markov chains, they're wonderful. They have closed-form solutions. They're all great, but closed-form solutions on noisy data isn't always that great. So we used this sort of clustering that was stolen from physics to sort of really find metastable states and infuse some robustness to this Markov chain that sort of overcomes noise in the data. So this is CellRank, and as I said, all the gory details are in like 80 pages of methods in the paper. But, just try and benchmark. It's very hard to benchmark, particularly one of the hardest things in our field is to find ground truth data. So here, thanks to Sam Morris, there was some cell tagging data, so we could look at what future clones would be. Now again, if you look at the velocity plot, nothing is going to reach success. This is cellular per proband. There's no stream plots here. But, we're working in high dimensional. We're working in sort of global Markov chain statistics. And so we said, okay, we know what clones, where they're going to go because we have their clonal information. And you could see we were pretty accurate. I mean, of course, at day 21, a final state was measured at day 28. We're super accurate here at 94%. But even at day 12, not less than halfway in the reprogramming, we were at 82%. So we were really happy with this. Of course, data in a dish is a lot simpler than real data. So we looked at regeneration. This is a regenerating lung. And we predicted a transition from goblet to basal that goes directly without any intermediary. And we indeed sort of managed to sort of validate that with some imaging data. Okay, so here we have CellRank. So we're going to apply CellRank to try and find cells of origin. Now we're switching back to our PDAC data. So we have one computational tool in hand, which I hope you understand a bit how and why it works, and you've sort of convinced it works. One of the best things it does is it finds very reliable and robust initiating states because of that Piron clustering that I showed you earlier. And these are the initiating states that it found. It found not one initiating states, but actually 4 initiating states. And 3 of them overlapped with initiating states that were proven through lineage tracing to sort of be it PDAC initiating states, sort of reconciling the field. It's like, yes, all of them are able to create, it's not that you're right or you're right, you're all right. It's wonderful in science when we're all right. Okay, so now we know the initiating states, and we want to know what are the dynamics. How do these initiating states sort of go forward? And here, we have another tool. We looked at RNA. Now let's look at chromatin. We actually had some bulk data at hand, which didn't force us to work, so we started from bulk data. And one of the things, again, you see in bulk data is that if you look sort of here in sort of the early tumor genesis, the 24, 48 hours under injury in KRAS, there's a mixture of like tannin and very advanced malignant states in the chromatin. There's a bunch of different chromatin modules. So we wanted to look at, okay, where are they expressed? And each of the modules are actually expressed. Even the chromatin isn't only open, the RNA is also expressed, but in different regions of the phenotypic manifold. So we sort of have different RNA states that represent different modules of distinct and mutually exclusive open chromatin. And here, is an example of like IL-33, which has been shown to be very important for advanced PDAC. And you could see that both in early injury and in PDAC, the peaks are wide open, but not in injury alone, not in KRAS, and certainly not in normal. So we felt, okay, we want to dig into it more. We collected single-cell data. This was before the multi-ohm kit. Some of these projects, especially projects that involve a construction of a new genetically engineered mice, they are a few technologies back, so we had to integrate them. So now we're going to take another quick computational segue of how we're going to integrate separately collected single-cell attack and single-cell RNA-seq. And so here, the idea is we're going to go, rather than the single cells are too sparse and hard to match. Clusters are too coarse and miss an incredible amount of heterogeneity. So we want a just right resolution of a metacell, which really represents a very, very tight thing. For anyone who's computational here, we're thinking of a single unimodal normal distribution. And basically, we're trying to fight very, very tight phenotypes where we believe the variation is technical variation in technology. So the first first time beta cells was articulated was by Amos Tanai, but his algorithm works for MARSeq, less so for 10x, and certainly not for ATAC-seq. So we developed our own version of it that's really robust for 10x, for RNA, and for ATAC. So why does it work? I always like understanding why an algorithm works. What C-cells does is it does archetype analysis, but unlike the archetype analysis that Uri alone told us to do on the count matrix that finds sort of extreme phenotypes, we're going to do archetype analysis on the matrix, on the affinity matrix, on the matrix defined by the edges. And what this does, it actually finds very, very tight archetypes, or these very, very tight cliques in the graphs of cells that are all very, very similar to each other, which is exactly what we want, very tight clips in neighbor graph. And the archetype is the single cell that is most similar to all the other ones. Again, exactly what we want. So this sort of archetype, those one slide ahead here. So this archetype analysis really throws this data into super high dimension, breaking it up into these really, really, really tight cliques in the neighbor graph, finding our metacells, or our C-cells metacells. Now, why is this so good to do? And I know that many of you analyze this type of data, so I'm going to show you how working at the resolution of metacells really gives you power. So again, we're still in our little computational segue. We're looking at hematopoiesis, and this is data really collected by multi-ohm, so we could do a direct comparison within each single cell of the peak accessibility versus the gene expression. These are tightly regulated things of master regulators of hematopoiesis. And within the same cell, the correlation between the master regulator and its targets, the correlation between open chromatin and the gene sucks. But when you aggregate information into metacells, there are about 100 cells per metacell, suddenly you get this beautiful correlation. You get the gene where it's supposed to be working here, erythroids, myeloids, dendritic cells way at the top. That's at the simplest statistic. Does the chromatin accessibility really match the gene? But, again, you want to know which peaks match it. This is what you get from single cell data, but look at the high resolution mapping of relevant peaks that you could get when you're working at the metacell resolution. all the way to inferring GRNs very accurately with all the known regulators and all their known regulatory logic. Again, here we're going to work at the cluster level. It is so coarse that it's so rough, it only picks up one regulator out of the 5 and in the wrong logic. So, this is C-cells, another tool into our box. Let's continue our adventure in PDAC. So now we sort of have a resolution that allows us. We have a sort of enough statistics. In fact, it has a distribution. We have about 100 cells per each. So we have a really good characterization of, the states that we see in RNA versus the states that we see in ATAC at fine resolution, and we can map between them and see that there's a pretty decent correspondence, as we see in this diagonal. We're happy the ATAC and the RNA sort of reflect each other, but not perfectly. We see a lot of this really nice off-diagonal stuff, which is like epigenetic potential to reach additional states. So let's formalize that and use that as a way to quantify plasticity. So what we're going to do is we're just going to build a classifier. Boring, machine learning circa 80s of a logistic regression classifier that will look at the gene expression and tell me what cell type I am. And, logistic regression is actually a great classifier. You don't need these deep monsters. Sometimes logistic regression even beats them, even though I'll use them when I need them, but not for simple tasks. Simple tasks often work best with simple algorithms. And so we have a great classifier to use gene expression to tell me what, cell type I am. But now we're going to say, but instead of looking at gene expression, we're going to use the very same classifier, but look at the open accessibility, how accessible that gene is, and ask, okay, can we predict it? In some cases, we can predict it really well. There's a one-to-one correspondence. We know what cell type is. There's no uncertainty. The classifier is, perfectly right. Some cases, it actually has open chromatin that matches a couple states, and we see a lot of that. So there is uncertainty in the classifier, entropy in the classifier, and that's exactly what we call plastic state. So looking at our data using this approach, we can see, as I said, some really nice one-to-one correspondences. These are the boring, non-plastic states that really map to each other, sort of these mature cell states. But actually, a lot of the data has quite a bit of heterogeneity in it. And for each of the cell types, we can actually measure a plasticity score, looking at that cell population. And what we see is that nest progenitor apex that we found earlier with cell rank is the most plastic cell. So the cell that was found by cell rank as origin is the most plastic cell. That is, it can reach, or its chromatin allows for, potentially the most amount of cell states and heterogeneity. This highly plastic state. Now, one of the nice things about quantifying something is that once you quantify something about a cell, like how plastic it is, you can correlate this degree of plasticity with other things. For example, you can run GSCA on this thing. And the top hit was cell-cell communication. In fact, the 10 top Go annotations were variations on the theme of cell-cell communication. Cytokines, ligands, kinases, whatever you want. 9 out of the 10 top ones were all related to cell-cell communication. So plastic cells have open chromatin next to genes that are annotated as cell-cell communication. So this sort of really hints that part of the reason these cells are plastic is that they have the propensity to receive send and receive signals from their environment. So that sort of led us to the next step. If plastic cells have a lot of potential to send and receive signals from their environment, how can we study that? So we zoomed in on the receptor and ligand molecules, and what we see here is this sort of beautiful block modular structure. So this is the sort of gene-gene correlation between all the receptors and all the modules and how well they're co-expressed. So we decided to find, so you see there's modularity here. We decided to find modules. This is sort of our algorithm calligraphy to sort of really understand communication between cells in a tissue. And one of the important things that we really wanted, a lot of the clustering algorithms is sort of one cell, one cluster, but we felt that we wanted not one gene, one cluster. We wanted to basically allow a gene to participate in multiple communication modules. So we specifically, as you could see, these modules are overlapping, and we found 7 communication modules. And we asked, okay, there's 7 dominant communication modules in this sort of pre-malignant KRAS, early injured pancreas epithelial cells, the acinar cells. Where are they expressed? Oh, this is sort of the final stage of our algorithm, which we'll go into a second. And what we found is that each cell actually predominantly expresses one of these modules. So it's not like basically each cell is largely expressing one of these 7 modules, and in fact there's a one-to-one match between cell type and module. So if we were to cluster the cells based on this communication modules or just your standard community detection on all gene expression, you'd get pretty much the same cell grouping. You'd see that these cells have like really high expression of each of these modules. Now this is very unique to the KRAS regenerating, this mutant KRAS regenerating context. So if we look at regeneration in the wild type, we see, this is sort of black, it's quiet rather than this colorful thing. We see only one module, the gastric module, open at much lower levels than in the KRAS mutant context. And actually, this module has gene expression of oncogenic KRAS, so it's basically, it seems like there is real KRAS acting in real regeneration that gets stuck and, sent on turbo in the oncogenic context, creating lots of new communication modules, none of which you see in the malignant PDAC. So there's a complete remodeling of the way cells can send and receive information in the oncogenic context. Remember, this is almost the same mouse. The only thing we did is make one mutation in one rare cell type inside the pancreas. Okay, so it needs to communicate with something. Under inflammation, there's a lot of immune cells, so we also profiled the immune cells. These are all wild-type cells. And we see that even though these are wild-type, there's no mutation here, they are dramatically shifted in state within 24 to 48 hours after injury. So in the KRAS mutant state, also the immune cells, the wild-type immune cells, are completely remodeled. So we could look for these communication modules in the immune cells as well. You could see that these communication modules, these sort of modules of bunches of ligands and receptors, not necessarily cognate, that all get co-expressed in the same cell. So this same cell has this program of a bunch of ligands and a bunch of receptors that are all co-expressed within the same cell, and that really defines how that particular cell can communicate with the environment. So let's harness that, and we're going to do something a little similar to cell phone DB, but we're going to harness the modularity and the robustness it gives us. First step is we want to know what's driving this oncogenic process. Why is the oncogenic and the wild type thing different? So we want to look at what's different. So we're only going to allow for edges that are different. So we can't, like if there's like a possible interaction that exists in the wild type, we're going to filter that out and allow for only interactions that are possible in the oncogenic context. Then we're going to use the modularity and look at communication between modules. So we're not, sensitive to that noise of one receptor and one ligand, but we have an entire module significantly talking with an entire other module where there's multiple cognate ligand receptor pairs that connect between two modules. One doesn't cut it. It could be that one is enough, but we want robustness. And then we do the statistical enrichment. Now, why was this important and why didn't we use cell phone DB to begin with? Because when you use cell phone DB, you get 99% of all possible interactions between all possible receptors and all possible ligands connecting all pairs of possible cell types. So, you get this fuzzball. This is the type of fuzzballs we saw a lot of in the early network meetings. Who was part of the field when we did? I mean, I see some of you. That were, basically an entire talks full of these fuzzballs. Now, yes, exactly. Some of us. Yay, me. And now fuzzballs are nice, but it's very hard to interpret and derive any biology out of this fuzzball. So rather than have 720 out of 729 possible interactions, calligraphy only gave 41 possible interactions. That's a sort of, this network looks like, hey, we might be able to interpret something out of it. So let's try and interpret this network. Let's look at what some of these edges mean. I actually like matrices more than graph when I want to quantify things. And so this is basically a crosstalk of each module, how it talks with each other module. This is sort of the significance of that link. And for each of these cell types, we can say, we can count how many links they have. What is their propensity to send and receive information? We have some of these cells that can sense their environment really well, or can remodel their environment by spewing ligands and cytokines into the environment. Now, these sort of really top two modules right here, the gastric module and the progenitor nest module, that was that sort of initial module that I showed you, that apex that comes up again and again and again using any metric. All these high plasticity genes that gave the enrichment that I was talking about are all expressed there. So it's a major communication hub. We see that these very same modules, they persist in advanced PDAC. So we're now looking at communication modules that emerge under injury within 24 to 48 hours. They persist in advanced, malignant, nasty, metastatic PDAC. They persist in multiple mouse models of PDAC, as well as in humans. So these modules, seem to be the real deal because they persist across mouse models, advanced stage disease, and even in human samples, which we pulled out of MSKCC. So let's try and dig into, one of these modules. Let's try and actually open. See, this is the sort of the most significant one between these two very, very, very big hub cell types. Let's open it up and see what it is. The gastric and the progenitor. You see that this module has 4 possible interactions between 4 receptor ligand pairs. But this is still, cells floating in a dish. Can we actually say that they're actually talking by showing proximity? So here we used SM fish. We measured 32 colors to get these 2 colors and 2 of these pairs, and actually saw, and now you see these modules lighting up with their cytokines. You see, the cytokines of the progenitor lighting up together in the same cells and regions, likewise in the gastric. And these are a few fields of view. But again, we could also do statistics and say the sending gastric module is in proximity of its receiving cells much more significantly compared to still gastric cells, but aren't expressing these cytokines. And you have to express both of them. So these are cells that express none or only one of these modules. They are much farther than those that express both these modules. So there's also a proximity here that's sort of validating calligraphy's link. We said, okay, but what we see is this really rapid, crazy rapid remodeling of both the tumor. I mean, this happens within 24 hours. Rapid dynamics, what does that, can anyone take a guess what rapid dynamics means? Feedback loop. We asked, is there a feedback loop? And there was one and only one feedback loop that we could find between the gastric, between the immune and the gastric compartment. The gastric compartment was talking with both Treg and ILCs. This module was in 2 types of immune cells. Again, you could see the richness of how many connections you have between these cells. It's not one; it's a bunch of connections. And, to sort of really dig into it, there was just an incredible amount of additional things downstream of these influences. So one of the things we found that was striking about this feedback loop, that if we could cascade and sort of build a link of this cell impacts this cell, and see how far this wave of influence can be as a cascade of events. This feedback loop impacted cells, which then impact the next module of cells, with impact the next module of cells. This feedback loop alone impacts 42% of the malignant epithelial cells and 76% of the entire TME. Again, to dig into it, you see this tiny, rare population of 3% of the epithelial cells expressing IL-33. You have a bunch of receptors here that can accept this IL-33. These very same cells that have the receptor for IL-33 are also expressing IL-4, which gets sent back to the gastric cells, to some macrophages, and really can do this dramatic rewiring. And again, this is very unique to the oncogenic setting. So if we're going to look back at our normal regeneration, quiet. Nothing's going on. IL-33, IL-4, none of that is being expressed here. Okay, so there's a prediction that an active feedback loop is really allowing me to get this immune epithelial crosstalk. So this is where we got delayed, and we built a brand new mouse where we can spatially, temporally impact IL-33 on will only in the acinar pancreas cells. So we're not even knocking down IL-33 everywhere. We're only knocking IL-33 down in the epithelial cells. And to see what happens, we see that first, 2 days after the immune compartment gets significantly remodeled, not much effect on the epithelial compartment. And, in the pattern stage, 3 to 4 weeks later, the epithelial compartment is significantly remodeled. And what we really see is that there's like a break in development. The sort of progenitor state gets blocked before it gets, into fully differentiated gastric and other states. And this is, again, you could see how dramatically, the progenitor state is enriched and the gastric state is really depleted. So there's a sort of developmental block in this cancer. And what I really like here is that it also completely validated our algorithm, because we have predictions of what cells are impacted by this and how far down the cascade. I mean, some of them were like 7 steps down. So we basically said, here is where you are, here's the predicted interactions, and this is how far down the cascade you are. These are impacted and really ordered in terms of the degree of change, in terms of how far down the cascade they are. And these are predicted to be not interacted. And this was like perfectly recapitulated by calligraphy's predictions. So again, really to the sum of the story, we have this beautiful story where the combination of a KRAS mutation and injury does dramatic chromatin remodeling. This chromatin remodeling, this change of epigenetic potential, opens a whole bunch of receptor and ligands, opens up their chromatin that remodels the way the cells can communicate and enables or rewires the cell-cell communication that enables, that sort of remodels the entire tissue and all the different compartments in that tissue. How many more minutes do I have? Because I have different... Because 5 to 7. Okay, so I'm going to go through the next story at a high level. So, what we do now in the lab mostly is look at all these technologies because, now we want to go in situ. I'm going to tell you very quickly about a project I did with Eileen Fuchs. It's published work. It's not supposed to be published. I know it's supposed to be mostly unpublished here, but it's stem cell. I don't think many of you have seen it. There's a couple of really nice ideas here. Again, we wanted to look at tissue interactions. 3D imaging allowed us to find a proximity between the lymphatic cells and the stem cells in the gut tube crypt, in human and in mouse. But, this is like 3 colors, so what are they doing? We want to get transcriptome information. Both in vitro and in vivo perturbation shows that the lymphatic cells phenotypically influence the stem cells. In fact, they hold them in a stem state. So when you remove the lymphatic cells, they begin proliferating. So the lymphatic cells are sort of inhibiting. And previously lymphatic cells were just considered to be like plumbing. They weren't considered to be doing anything interesting. So how is that happening? So for this, we looked at sort of Visium. The problem with Visium is it's like these big, ugly spots, and it's hard to look at the resolution. So how do we integrate single cell and Visium? Now, most of the ways to do that work on regression, linear regression, which are pretty crappy. And what we decided to do is go Bayesian all the way. And this is work by Tinney. And the idea is we want to explain the data. And the trick is not only to explain the cell type fraction, but also we want to reconstruct the entire count matrix. for each cell type in each spot. So we're inferring more, but by forcing the algorithm to infer more, we're actually enforcing more coherence on the system. So by really demanding not only to get the vector of cell type fractions, but the entire expression matrix for each cell type in each spot, which sounds crazy, we're actually getting a lot of power. And where can we do this statistics? We have to get the power somewhere to do this crazy inference. We're actually pooling information across all the spots. So we are inferring per spot, but we're pooling information per spot. So we use the RNA only as a prior. So, we could actually change what's going on and develop new types like in a cancer setting. Now this really gives us a lot of additional power. Let me show you how. It's again very hard to get a ground truth, but we had our friends in Elaine's lab build a ground truth. We were interested in the lymphatic cells. We were interested in the stem cells. They're rare cells. And, in a benchmarking, we're up here on top, this red thing, on how accurate we are against, stained ground truth data, and we're the best. Now, some of these other methods look pretty crappy. They're not crappy all around, but they're crappy on rare cells. Most machine learning algorithms are really designed to get average. Most of our metrics look at average, but all the interesting biology is in rare cells. And remember I said we could pick up specific cell types? We could pick up cell type specific expression. So we looked at cell types that were in the same spot, that had different levels of the same genes. And again, using a ground truth, we could say that we even got the count matrix correct. Okay, so now we can trust it. We go back, and we're still stuck with these big, ugly spots. And there's like 4 spots per crypt. How are we going to get high resolution? Again, using the stereotypical structure of the gut tube. When you're working in normal biology and not messy, ugly, disgusting cancer, there's beautiful structure in biology. So we're actually going to look at the cell type fractions, turn that into a vector, and do standard, dimensionality reduction style diffusion components. Here we're using fate. And now we're going to get basically high resolution. We're going to use 4,000 spots, place each of these spots on a pseudospace along the crypt, and quantify in a complete continuous pseudospace for each spot up the crypt what are the cell type fractions and what genes are expressed in each cell type as you go up the crypt. This allowed us to really pick up relin as something that's expressed only at the very bottom of the crypt and not at the top of the crypt in these lymphatics. So this is at the bottom of the crypt. You see it goes down. It's uniquely expressed only in the lymphatic tissue. Here's a validation of its expression in actual immunofluorescence. And we could validate with functional perturbations that relin knockout completely phenotype all the experiments that we did by co-culturing lymphatics or knocking lymphatics in vitro and in vivo, showing again how powerful Bayesian integration to get accurate count matrices and using the stereotypical structure to get high resolution allows us to really fine map a cell-cell interaction in real tissue. That was just like where we need to go when we're mapping now in situ. Finally, we have a lot of fun stuff. My lab always has endless postdoc positions. HHMI makes me rich. What you guys might not know is that we have a single cell innovation lab, which has both experimental and computational staff positions, well paid. So if you want like a comfortable, fun, innovative environment, there's a lot of them. And we're towards the end of our faculty search, but also the program is expanding. And finally, lots of great people. Cassandra did most of the work together with Dorena, now at IRB. Kinney worked with a power team. I mean, having one computational biologist analyze the data of 3 stellar experimentalists is a joy. And then there was a bunch of other great people in the lab that developed the methods. And this is drunken Shakespeare. If you ever have an opportunity, go for it. And thank you very much.
Thank you. Questions?
Thank you, Dana. Beautiful stories, plural.
I told one for real and one on speed
But maybe
mode.
on the one for real. So it's all in the context of the G12D mutation,
Yes.
KRAS mutation. Like, how fundamental do you think that is to actually PDAC? Do you think it can be initiated in the same way outside of that context?
So first of all, PDAC is one of these diseases where almost all PDACs have. It's like
exactly.
First of all, 95% of the PDACs have that mutation.
It, yes.
So
does it start that way? Or does
so
it,
they believe that, again, if you believe that the field, there's they believe it starts that way. People, I mean, certainly the mouse models recapitulate the human behavior, but also there's a lot of tissue taken from sort of pre-malignant people with pancreatitis, people with sort of propensity for PDAC, particularly now with the whole, if you're Ashkenazi and have BRCA2, you're absolutely screwed, because not only do you get breast and ovarian cancer, but you also get pancreas cancer and prostate cancer. So there's some BRCA-related studies where they already see in some of these patients some KRAS mutation. So in some, again, very limited, nothing that I would say definitive like in the mouse, but we also have some human data showing that there's, tissue that is otherwise normal with that nasty KRAS in normal-looking pancreas in human beings. So people think it initiates from there. Regardless of that specific mutation, I do believe, and we're working now in other contexts, and we see this similarly in colon as well, that the concept of a mutation altering epigenetics, altering plasticity, and altering the way you communicate with your environment, I believe that generalizes beyond KRAS, generalizes beyond pancreas cancer, and it's still early days, but we're seeing a lot of examples of that.
You answered my follow-up question.
That was fun. Thank you. So do you see opportunities for disrupting your crosstalk networks in useful ways, and how would you figure out exactly where to start if you've got 4 receptors and 4 ligands, and how would you?
So the IL-33 mutation was sort of one gene, and that was enough to really block it and block the progression, and even the pannins look differently. So again, I don't know if this is enough to block it forever. My suspicion, though we didn't run through it, is cancer would always find another way. But IL-33 alone, and it is druggable, is enough. The main thing is, how do you identify the human beings that need to be drugged with IL-33? I do think that it's possible to drug early, but you just have to find the people. And as you saw, that network is complicated. You block one, you slow it down, so I'm not sure you can entirely stop it. My attitude is if you find anything suspect, just cut it all out, but that's, But I do think there's value in understanding the process. And yes, it is druggable if you figure out who to drug and when. I fear that cancer will find another way. Very innovative, unfortunately. Everyone's tired.
And actually, yeah.
Yeah, this is a bit of a higher level question, but inspired by what might have been an offhand comment, where you said that for some models, right, like just asking, disease or not, or tumor or not tumor from single cell data, we'd say is a simple task that logistic regression can handle better than much anything else. I just wanted to ask you, what do you think is like a complex task for a single cell that would demand like a more sophisticated model versus what people would conventionally try to throw their biggest thing at that would otherwise be really simple?
So the places where first of all, deep learning works for anything imaging because, if you have images, even your basic pre-processing tasks. But, one example is data integration. So, we found that there's a lot of data integration that really works well when you combine the two modalities. we found that really, if you do it correctly, there's a good way and a bad way to do deep learning, but if you do deep learning right, you can integrate, for example, MERFISH and RNA in much more powerful ways than other integrations. Another direction, I mean, one of the great things about deep learning is it is nonlinear. It's not interpretable, but, a lot of biology is nonlinear. Another way we're using it is, again, trying to map the craziness of gene regulation. So again, the deep learning models that try and say, let's go beyond our boring, linear, simple PSSMs, and let's, run deep learning on trying to learn higher order structure in the genome. That's probably the biggest bang for your buck in terms of where deep learning has had the biggest impact.
Thank you.
So, right here, Dana.
Okay.
Right here. What do you think could be the biological function of the plasticity, the epigenetic plasticity, the plastic potential that you observed? And could this also play a role?
Well, I showed sort of two examples of that. One is during development. During development, there is so much movement. I mean, people don't really appreciate it, but when you're actually a developing embryo, you combine massive proliferation with massive movement. It's not that, it's very robust in the end, but where each cell will go in these sheer physical forces, as you saw how those cells just got sucked into the gut tube where they weren't supposed to be. So this plasticity allows you to overcome that sort of uncertainty of where you'll end up with all this crazy amount of movement after all the proliferation and development. And so really to allow the tissues around you to pattern who you're supposed to be based on your neighbors. And in regeneration, I mean, that's the whole idea of regeneration. Something's injured, you have to sort of fix it. You have to put all the other cell types there that need to be there. You have to sort of rebuild what's broken. And so regeneration, and again, we saw how this was using mechanisms of regeneration, is another place where this plasticity is needed.
And does the plastic potential correspond with the potential to regenerate?
So we see we see an incredible degree of shared mechanisms. Basically, cancer hijacks and warps. I mean, I showed you the example of that, how the metastasis really hijacks that lung organogenesis program. We see, the cancer, decades ago was called the wound that doesn't heal. It's very clear that cancer doesn't reinvent the wheel, but it hijacks and twists and makes aberrant programs that were originally derived for development and regeneration. And because it sort of alters the way they communicate with the tissue, but yeah, it doesn't reinvent the wheel. It actually really harnesses those programs that were there for good physiological purpose originally.
Sure. I was going, so yes, I admit I missed a good chunk of your high-speed
Sorry.
talk. No, that's fine. But I
tried to make the first part at least reasonable paced.
It's not a computation background. How much role does this potential have in complex diseases and with regard to being more sensitive to environmental stimuli and developing asthma and those kind of things?
I mean, again, these plasticity is highly connected to sort of more stem and progenitor cells, but they also play a role in a lot of more advanced disease. Certainly, plasticity may be less, but cell-cell interactions, for sure. I mean, a lot of these diseases are very inflammation-based, and that the type of interactions and the aberrant interactions between inflammation and immune system and epithelial tissue. So cell-cell interactions, I think, are driving a lot of disease. Plasticity, a good chunk of them.
Hi, Dan. This is Fei-Chung Chen from Cleveland Clinical. So my first question about your cell range, right? So you really show very fabulous biology discovery from your cell range approach, right? My question is, do you think you can integrate, this kind of, omics data with the network data, like a protein-protein interaction network, use the autoencoder approach to further improve your cell range, tools? Do you think it's possible?
So, I mean, in a way, I used protein, again, a different type of protein-protein interaction network, but calligraphy was heavily fed and heavily uses known cognate ligand receptor pairs, and that is a type of protein-protein network interaction that was critical for calligraphy to sort of build its interaction. So that's an example where known protein interactions were infused. For me, if we're talking about the protein-protein interaction field, which I know a lot of you deal with, in cancer, it's really altered. It's very context-specific. So what I've noticed in the cancer world is the protein-protein interactions you're building in your own world, they're different across individual patients. I mean, you can't even build me one for PDAC. So particularly because of the way I think they're so context-specific and so altered in a cancer patient-specific way, that's not where I'm personally going. But for other diseases where maybe that protein-protein interaction is a little bit more stable, I'm sure that there's ways to go.
So my second question is, you really show multiple tools, right, applied to different biological questions, but most of the tool is really data-driven tool, right, from supervised learning, unsupervised learning. Did you have any insight or any suggestion about like deep generative model maybe also can apply to this field as well, right, to do really some like a novel discovery based on some, very beautiful, or fabulous example?
So, it wasn't a deep generative tools, but a lot of what we found was very discovery based and driven by sort of unknown. So it was unstructured. I gave earlier examples where some deep tools, work really well, and we have some in the lab, but you don't have to be a deep generative tools to be data driven and to discover new things. Discovering new things, just means not forcing too much prior on your data.
Hi, Dana. This is Xiang. As a student from computational background, I am wondering, at certain stage, what brought your team's attention to look into the epigenetic basis of cell state instead of keep mining into other existing genetic data or omics or like drug perturbation?
So I'm very interested in gene regulation. So one is very, very clear, and I was always interested in the epigenetics layer to sort of understand gene regulation. And actually, if I want to tell the story in true form, we originally collected the epigenetic data to reconstruct the gene regulatory network, which was less stable and robust. But if you want to think of plasticity, if you want to think of how a cell can change its cell states, if you want to think of, once we realized that there's plasticity and there's these cells that seem to express multiple programs, we thought that could very much be encoded in the chromatin. So then we took a different direction because we collected data and in the meanwhile was analyzing, and then we realized, hey, we have a really strong plasticity angle and story, and one of the clear sort of mechanisms that could be driving plasticity is chromatin. And we saw also that the chromatin was dramatically remodeled. So we sort of followed the chromatin remodeling and plasticity angle, which is easier and more novel and interesting than in gene regulatory. I mean, we also do gene regulatory networks by combining ATAC and RNA. That was the original reason we sort of started looking at the ATAC, and then we followed the data and the biology of the story and went a different direction.
Thank you. Interesting story. Thanks a lot.
So a minute ago, you just said that the protein interaction networks of different patients are different. Is that because cancer mutates the gene and therefore the sequence of the protein and therefore its fold? So it has different interaction partners?
I think, again, a lot of these networks are very signaling based. So I'm not talking about the protein-protein interactions that sort of get, physically bound. But a lot of what plays a big role here is signaling. So what's expressed, different arrangements of scaffold proteins. There seems to be a lot of sort of, particularly, for example, in the EGFR pathway and its family, there seems to be a lot of feedback loops where you sort of shut down one. I mean, what happens with all these signaling pathways is just feedback loops that cause re-expression of things, that cause rearrangement of what goes through what. And because it's signaling and not like this sort of physical hard protein-protein binding, I don't think you need too much of a mutation to cause those changes.
So it's more what's expressed as opposed to the proteins changing shape.
What's expressed and how they're organized into this sort of scaffold and what sort of where the predominant and I also think, and we haven't, there lacks good models because I think a lot of the biophysical modeling of signaling networks sort of takes a too narrow view. But I think in signaling, it's a lot of not just like the biophysics modeling of one network, but once you have sort of flow through one network, you waste another network. So what is sort of, you're thinking of it in terms of network flow, more the type of things that people have been doing in metabolic networks and flux balance analysis. I really think that some of those methods should be applied to sort of protein signaling methods to try and get more global impacts of one network on another.
Final clarification, what do you mean by scaffolds?
They have to sort of come together, and there's this sort of different dimerizations of these proteins, and you do see different dimerizations when you actually go to sort of more mass specu-like data.
Hi, I was very struck by the difference you have between your metacell regression values with the chromatin structure and the single cell values. Huge deconvolution of the signal there.
So I took a fast thing of the cell, but again, that's sort of online in bioRxiv, probably be on Nature Biotech next week or something online. The thing is that single cell ATAC-seq is sparse like crazy. So you have about 200,000 possible open peaks. You have about 2,000 actual reads coming from each cell. And the noise levels in single-cell ATAC-seq are higher, so about 1/3 of them are garbage background. So you have 1/3 garbage, 2/3 real signal, and only 1,400 reads covering 200,000 peaks. It's crazy. And when you aggregate 100 cells together, that's where sort of double your signal. One, that completely clears out the background because it wipes out the background noise. So that third of noise just gets wiped out, and then you have 100 times more data, and suddenly you have signal.
So you think that difference is
the aggregation? Yeah, the aggregation, which cleans the noise and gives you 100 times more signal.
You're saying that noise is mainly technical because of the nature of the
nature of worse noise to signal ratio than the bulk, and just the sparsity. I mean, it's crazy sparsity.
Thank you.
And maybe with that, we should thank our keynote speaker again.